Good morning. Hey guys, and welcome to our lesson on phloem. The last couple of classes we have been talking about the transport of water going through plants, and we said that it was kind of a tricky business. In fact, it is probably the world's most complicated and intense straw that has to bend the laws of physics in order to be able to actually transport water all the way up a plant, right? But what about the sugars, right? Water is, of course, the primary problem. If you can get over the problem of trying to handle your water need, then you're okay, right? Because all life needs water. But then the second thing is, of course, how do you handle the transport of your food? And it's not so easy in plants, right? With heterotrophs like us, we can just walk up to something and be like, hello, I would like to eat you, nom. And then we eat it, and then it goes straight through our wonderful unidirectional digestive system. And after a couple of hours or a few minutes, if you're talking about Taco Bell, it comes out the other end, and then we're good to go, right? But with plants, it's not so easy. They produce the food within themselves because they're autotrophs, and so within their photosynthetic organs, they produce all of this food, and the food has to go throughout their body to where it's needed, but they don't have blood. They don't have a digestive system. They don't have a stomach. They don't have intestines. They don't have a mouth. They don't. Okay, so how does food get transported through plants? How do they do it? And especially if you think about their long bodies, I mean, think about a 100-foot long tree. That food has to be able to get from the leaves at the very top of that tree all the way down to the growing roots of that tree, and it has to happen in a timely manner, right? It has to happen fast enough so the tree doesn't starve while the food's on the way from its leaves down to its toes, basically, right? But so how does this achieved, and especially how is it achieved in a bidirectional flow? Because if you think about it, the tree is probably going to be sending resources down to its roots, sure, but what if those extra resources are needed back up in the stem, right? Because it's growing both ways. How can it transport sugars both ways through its body without a digestive system? That is our puzzle for today. Now, you guys have read through this section here at this point, I'm guessing, but I'm bringing it up here because I want you to watch this video. And I know in the video it's a little bit evil of me because I'm having you look at how the trees are using sugars as an adaptation for the cold. But the big thing I want you to focus on in terms of this video is when it talks about the chemical potential. Okay, the chemical potential is very much like the water potential that we talked about in osmosis, right? We said that if water always goes from a higher water potential to a lower water potential. And I just want to remind you of what that means. Essentially what we're saying is that water always flows from a purer water to a less pure water. When I say less pure, I mean that it's mixed with solutes like sugar or salt or something else like that. And the reason for this, right, this osmotic flow, is that the water that is within the purer water is going to have less of a resistance or binds or any kind of chemical reactions with the things that are going on. And it's attracted to the chemical reactions, right, the chemical properties, the bonds, the charges within the sucrose and all the other things that it could be mixing with. And so there's this natural passive osmotic flow. It takes no energy for this to occur. It's because the energy potential, right, of the water that is pure is higher when it is in the presence of solutes, so it's going to follow that potential, right? Because we know that atoms and molecules such as water are going to want to make bonds if it can get them down to a lower energy state, and it can if it is binding to solutes, so it naturally wants to go in that direction. So when we talk about chemical potential, it's something similar. We talk about the higher chemical potential that is within the liquid water versus the ice that's forming outside of the cells that's talked about in this video. The water is going to want to flow outside the cells towards the ice. So in the same way, I want you to imagine, what if the sugars produced by photosynthesis in the leaves, which are immediately converted to sucrose, have a higher chemical potential than the forms of the sugars that are actually being used in the plant tissues, right? Okay, so in other words, the factory that's producing the energy is creating an energy source that wants naturally to be used, and the places where it needs to be used has much lower energy. And if energy naturally flows from high to low, how could you translate that into sugars flowing from the photosynthetic organs to where they're needed in the plants? That's the general concept. So I want you guys to take a crack at that here before you really start hearing what I'm about to say next. But I want you to start with that idea, and we're going to build off from there. So go ahead and try out the discussion board post, pause the video, and then come back in a moment and see whether or not your hypotheses were correct. All right, now let's actually talk about what is going on. So The transport of sugar in plants is known as translocation, right? We're going to talk about translocation and then phloem loading, which is a specific part of the process of translocation. But we're going to start with the general concept of how sugar flows through plants, and it flows from sources to sinks. And if you're wondering about this, like what the heck does source and sink mean? Why are we talking about sinks? This is a plumbing class. Well, we're talking about plants. It's basically a plumbing class. But sources are where sugar is made. That's your photosynthetic tissues. So we're going to be talking about, all this wonderful palisade mesophyll cells and spongy mesophyll and all that stuff that's going on in that leaf tissue. But that's going to be the actual places where we have the chloroplast, we have the photosynthesis happening, the Calvin cycle, carbon fixation, the creation of glucose molecules and other carbohydrates that are the essential food sources for plants. Sinks are going to be where you're putting all that energy, just like people talk about, sinks for your money. My car is a money sink. They mean that their money is constantly going into their car because they have to constantly fix it, right? And that car is basically a pit. They're sinking all their resources into it. So a sink is going to be where this energy is needing to be used. And as you can imagine, sinks include fruits or seeds, right? These are things that are going to require energy because they're either going to be storing energy up for later or providing food for their seeds to grow into offspring, which is, hopefully what we all want to have happen to our lovely little tree seedlings when they go out there and not just die without any food to eat, because that would be sad. Little orphan seeds standing there being like, please, can we have more sun? Okay, and then we have roots, right? So the guy in shoots, obviously. So the plant's going to be growing, right, out of all of these different places. So it's going to need energy for its growth, right? And so that is what we are going to be using. So we're all going to be going from source to sink. That's the big thing. The big thing to also understand is that sources and sinks can be in all sorts of directions in relation to each other. It's not just from the leaf down to the sink in the plant. It could be from the leaf down and over and up, because sinks and sources can flip-flop in the tree depending on what is photosynthesizing and what is growing. It's this constant thing, right? If you think about a leaf that's growing, it's a source because it's photosynthetic, but it's also a sink because it's growing and it needs energy. So it becomes complicated when we talk about where stuff is going. But essentially, the organic molecules that are transported via the tube system, these sugars, this is what we call active translocation, right? And it's going to be the active loading of solutes into the phloem, right? And then using water to actually create a pressure and osmotic gradient to move these resources around. Let's talk first about the phloem structure before we talk about how it moves. Phloem structure is comprised of several different things, and you'll notice some immediate differences from xylem. One, phloem is living, which is very different than xylem, which is dead. It was a xylem tube and it was dead. Phloem is comprised of sieve elements and companion cells. A sieve element is essentially just going to be sort of like the drain in your sink. It's going to have holes in it, it's going to let stuff through, so it's going to be a partial barrier. And you're going to notice where this actually comes from is in the ends of the plant cells. Basically, phloem, just like xylem, is a bunch of cells stacked one on top of each other that make a long tube. They're all connected. But this time they're living, and there are small holes punctured in the cell walls on the ends in order to allow a flow all the way through. So basically it's a pipeline, right? But it's not just an open tube like xylem. It has little holes where the cell walls go, right? And so that we can see this is where the stuff will actually flow through. And then it also has companion cells. Companion cells are going to be these cells that are over here on the side, and those are going to be living cells that are going to be facilitating the bidirectional flow, which is another thing you should notice. The arrows are going in two directions, whereas in xylem they were only heading in one. And this is because our sources and sinks can change places and flip-flop, and that's not true with water flowing through xylem. Sieve elements have no nuclei to maximize space, so you can see that they're just basically empty pipe tubes, whereas the companion cells have all of the nuclei and everything else you would imagine, along with a highly folded membrane to maximize surface area for more reactions to occur. We're going to talk about that in a second. And then there's also plasmodesmata, which are going to be those small little tubes we talked about in the xylem that are going to allow for a flow of materials from one cell to the next laterally, between cells that are, I guess, parallel to one another, kind of stacked next to each other. And that's going to allow for those elements to flow through their collective cytoplasm. Here's a nice little diagram showing a wonderful comparison between the two, right? So we have phloem, transports organic molecules. We have these sieve elements instead of this open area right here. There's two-way movement, and they're living, versus the xylem, which is transporting water and minerals in a unidirectional flow with no end walls, one-way movement. Outer cell walls are not living. Right? Okay, good. Okay. Let's talk a little bit about companion cells. So why the heck do we need companion cells? Why does it have to be living? Because if we are going to be transporting things bidirectionally, we are going to need to sometimes use active transport to transport stuff against its concentration gradient. It's just going to happen. You can just see how it's going to happen because sometimes you're going to have your concentration gradient flipped and suddenly you're going to need to reverse the direction of the flow. How are you going to do that? Well, you're going to have to reverse your concentration gradient, and the only way to do that is to pour energy into active transport. So companion cells support phloem by providing metabolic support. So essentially they're going to provide all of the mitochondria, which are going to be the powerhouse of these cells, creating all of the energy in order to be able to power this movement through the phloem. So the companion cells are going to move material in several ways. They're going to have the plasmodesmata for just the typical flowing of stuff through passive transport or what have you through the cells, just quick and easy. Very small things can flow through those. And then by actively pumping materials from within the cell wall space of the companion cells. And this is, of course, what we were talking about kind of before in the xylem. This is the apoplastic pathway, whereas just traveling through the plasmodesmata is a symplastic pathway. Apoplastic because we have to go through a cell wall and actually go through a transporter protein, and symplastic because we can just go right on through. We don't have to worry about traveling through any surface. Now, what is phloem loading? Okay, so phloem loading is the apoplastic side, okay? And so I want to go back to this image actually to be able to show this. And actually, no, I'm going to use the one from the textbook. So if you go into your textbook, and this is section 9.2, there's this wonderful little diagram right here. And this shows what we're talking about here, all right? So if we look here, this is where our phloem is right here. And it's a little bit difficult to see. So I'm actually going to use a combination of these two. Whoops, all right, sorry, I went to the wrong thing. Okay, all right, so when, look at this one. Okay, so here's our phloem tube, right? And here's our companion cell. Okay, our companion cell is going to have to pump sugars and other things into this phloem tube to start setting up a concentration gradient so that there's more sugar in the phloem tube and it'll start flowing where it needs to go. Okay, now this is a top-down view. I'm going to then describe this as a side view. Okay, so here's our phloem and here's our companion cell. Okay, so it's the same thing. This is looking at it from the top. This is looking at it from the side. Okay, so we have our phloem and our companion cell, and then these are all the other supportive tissues, right, that are around the phloem. Because when we look at these vascular bundles, right, it's a bunch of these tubes side by side. So that's kind of what we're looking at, but from the side, right? We're looking at it from the side. So here's our phloem. This is where we need all of our sugars to go, right? So we have our photosynthetic stuff, and here's the symplastic pathway. The sugars flow over here, but then we need them to go ahead and actually go into where we want to send them. Now, in order to do that, we are going to have to set up some energy in our companion cell. Our companion cell's job is going to be to pump these sugars where we need them to go. Okay, and so the companion cell is going to have tons of mitochondria, and it's going to be producing a ton of ATP, and it's going to start producing a concentration gradient. So we're going to see that right here. It is going to start to pump hydrogen ions into, out of the companion cell into the phloem. Right, or excuse me, out of the phloem and into the companion cell. And what this is going to do is it's going to start to set up a hydrogen ion gradient inside of the companion cell. Now, you'll notice that this is very much like what we looked at when we were looking at photosynthesis. When we were looking at photosynthesis before, and especially in the light-dependent reactions, we had to set up that huge hydrogen ion gradient in order to be able to have those ions flow through ATP synthase and power ATP synthase to produce ATP. So we're doing something similar here in that we're pumping a bunch of hydrogen ions from the empty phloem into the companion cell, and the companion cell has all the sugars, by the way. We need to take those and put those into the phloem. So we have all these hydrogen ions building up inside the companion cell, and that is going to make a concentration gradient that's going to want to make those hydrogen ions flow back out of the companion cell and into the phloem. But when they do, they flow through something called a symporter. A symporter is a transport protein that when it transports one thing, also transports another. So the symporter is powered by the hydrogen ions who naturally want to flow through it, but it's also carrying these wonderful sucrose molecules. Sucrose is, again, the form that sugars take after it comes out of photosynthesis. So basically, in the companion cell, we have a bunch of these sugars that are produced by photosynthesis, and they're going to be converted into sucrose for transport, and that sucrose is going to then be transported using the symporter in the hydrogen ion concentration gradient so that it goes into the phloem. Okay, so once again, hydrogen ion gradient powers the symporter. We have the sugars flowing from the companion cell into the phloem, and boom, we have our phloem going into our, we have our sucrose, our sugar, going into our phloem. So this is how we actually get the sugar into the pipes. It doesn't just start in the phloem, it has to be transported into the phloem from all of those cell tissues that are coming from the photosynthetic organs. Once once we get the sugar into the phloem, now we can go ahead and start using water potential. What do I mean by that? Okay, so now we have our sugar in the phloem. So here's our source, here's our companion cell. It's gone ahead and pumped that sugar into the phloem. And now we have a much higher concentration gradient of solute here in the phloem than we did water. Okay, so now that we have a much higher concentration of solute, what's going to happen? Well, the water that's flowing in the adjacent xylem tube that's right next to the phloem is going to be like, whoa, hey, well, I'm at high water potential here, but that is some low water potential over there where the sucrose is gathering. So the water is going to naturally want to flow from a high water potential to a low, or from, if you want to think of it this way, from a low solute concentration to a high. So the water is going to naturally want to flow from the xylem across those plasmodesmata into the phloem. Now what's going to happen? As soon as the water hits this sugar in the phloem, it's going to start to build up a bunch of pressure because there's not a lot of space for stuff to go. Water is incompressible. You cannot compress water. So water is going to get in there and it's going to be jamming up into this tight space with all the sugar being like, all right, this is great, right? Like people going into a concert and then they're going to have nowhere to go but down the phloem tube, right? Towards the area of lower sugar concentration, which is of course going to be your sink because your sink is using up all the sugar. So your sink is down here and the sink is using up all the sugar as it's growing. Maybe it's a root or something like that. And it's constantly using up those sugars. And as that happens, the water is naturally going to be like, I got to get out of here. And it's going to flow from the jam-packed source down to the much less pressure sink. And it's going to, while the water flows, of course, deliver the sugars that are inside the water so that they can make it down to the sink. So phloem loading, in terms of the apoplastic pathway, all has to do with us setting up a concentration gradient at the beginning using active transport and then letting the natural osmotic force take hold with the pressure differential to actually transport stuff down the tube to where the sink is going to be, which is really ingenious if you think about it. It takes a little bit of energy at the beginning, but we get a massive payoff there at the end. Now, let's talk a little bit about the phloem unloading. So once the sugar actually makes it down the phloem tube to the sink, it is then going to need to be transported into the actual sink itself. So this is actually going to be pretty easy because the sink is using up a ton of the sugars already. And so what's going to happen is we are naturally going to get this passive flow of sugars from the phloem into the cells towards the sink where it needs to go. Right, so our organic molecules are naturally unloaded from the sucrose laden phloem into our sink. This is going to cause the pressure to drop and the water potential to return to normal. The water is going to go back to the xylem, not carrying any sugar with it. Remember, the xylem is just basically pure water. And this drop in pressure is going to make sure that the concentration gradient is maintained. We're going to have low pressure at the sink and high pressure at the source. And we can see that the direction of mass flow can also be reversed according to needs. So let's say that we no longer need any more energy here in the sink. We're good to go. We can then use ATP to go ahead and do a similar thing, push all the sugar back into the phloem, just like we did at the beginning with the companion cell, and then it will flow towards the next nearest sink. So in summary, we have our companion cells that are going to be next to our source for photosynthetic energy. They're going to actively transport sugars into the phloem. Water from the xylem is going to be naturally attracted. It's going to flow down using osmotic force to draw those sugars down to our sink, which are naturally going to take in the sugars. The water will return to the xylem. The pressure will remain low near the sinks, and we will have a constant flow from source to sink. And again, if the sink and the source swap places, doesn't matter. We can just actively transport back into the phloem, and the water will naturally take it from an area of high pressure or high solute to low solute and low pressure, which is exactly where we need to go. So that is, in a nutshell, how this works. Now let's talk a little bit about, and we're going to look at a quick example here before we take off there with this, but, and actually, you know what, I'm going to go ahead and show you that example right now before we move on. So this is a database question that I have given you guys to solve, and you might look at this and think, okay, nope, that's it, I don't really want to do IB anymore. Thanks, Mr. Garner, but that's fine. I don't want to look at this. This is terrible. But let's go ahead and break it down. Okay, so I know this is scary, but all this is asking asking me to do is describe the forces that are inducing water or sugar to move from one location to the next. Okay, and I want you to go ahead and take a look at these little symbols here. These symbols are not demonic symbols, or they might be, but right now they're not being used as such. And they basically are just talking about potential. So this weird little Greek symbol here means potential, and the little letters next to it mean stand in for what it's talking about in terms of potential. So this is water potential because it has a little W. This is pressure potential because it has a little P. And this is sucrose or solute potential because it has a little S. So let's go ahead and take a look at what's happening here. Here's our phloem with our companion tubes right here, and then this is our sink, and here's our source, and here's our xylem with the just unidirectional flow of water. Okay, it's asking us to explain the movement of water from point A to point C. Well, let's take a look at our potentials over here, okay? And I want you to remember that potential always flows from high to low. So let's take a look at what's going on. Our water potential here is 0.8. Our pressure potential is 0.7, and our solute potential is 0.1. In other words, we have a decent amount of water potential, pretty great actually. We have pretty high pressure, and we have pretty low solute potential, which makes sense because we're in the xylem. We shouldn't expect there to be a lot of sucrose floating around in the xylem. Now let's look over here at the phloem at C. We have a water potential of 1.1. Okay, so that is a lot higher there. We are going to have a pressure potential of 0.6, right? And we're going to have a solute potential of 1.7. Okay, so in other words, we have a crap ton more solute over here, right? And we are going to want to flow towards that solute. All right, in other words, we have our water potential over here is going to be much higher, much closer to pure water. And I want to remind you, sorry, this might be confusing to you because you're looking at this and you're like, what the heck over here? Okay, remember that the closer you are to 0, okay, that is going to be your higher water potential, okay? And that might seem counterintuitive to you, but 0 is the highest water potential because it is pure water, okay? And so when we're getting higher with our numbers here, we're actually getting away from a water potential, right? A higher water potential. So here we are going to want to flow from a high water potential at 0.8 over to a low water potential at 1.1. Okay, so the water is going to flow from the xylem over here to the phloem. Okay, and now that we have the water over here, we're going to get a crap ton of pressure starting to build over here, right? So our turgor pressure is going to start to become huge, right? And that is going to be because water is incompressible. It's going to naturally want to flow to a lower pressure situation. So now let's go over here to explain the movement from point C to point D. So if we look over here at point D, we can see that our water potential is 0.4. Okay, so closer to pure water again, which is, so it's a higher water potential down here, lower again over here. Remember, the farther away we are from 0, the lower our water potential gets. Over here, we have a pressure potential of 0.3, and we have a turgor pressure of 0.7. Okay, or excuse me, a solute potential here of 0.7. So if you're looking over here in terms of where the water is going to flow, we have a much lower water potential here and a much higher water potential here. It's closer to pure water. So the water is not the force that's going to be causing the sucrose to come down here. That just wouldn't make any sense at all. Okay, so let's take a look at the pressure. Okay, well, the pressure up here in terms of, and you got to look really hard to be able to see it, but the pressure here from 0.6 to 0.3, okay, so the pressure here is going to be a lot higher than it is down here. Okay, so we have high pressure to low. Okay, so that is a potential, and we're going to have a high solute potential here at 1.7 versus a low solute potential here at 0.7. Okay, so there are two forces that are going to be inducing the sugar to flow down here. Then we have a higher pressure potential here and a lower one here, so the pressure is going to be causing it to flow, but also we have a higher solute potential here because we have a heavier concentration of sucrose and a lower solute potential down here, so it's going to want to naturally flow down here. So pressure and solute potentials make it flow from C to D. Okay, and then finally, let's explain the movement of water from point D to point B. So if we look at point D, or excuse me, point B, we have 0.6 for our water potential. Okay, so 0.6 versus 0.4. So actually we have purer water over here than we do over here. Okay, we have a pressure potential over here at 0.5, and over here we have a pressure potential of 0.3. And then over here we have a solute potential of 0.1, and over here we have a solute potential of 0.7. Okay, so in other words here we have a much higher solute concentration to low solute concentration, so that could make our flow go over here. But remember, we're talking about the flow of water, so water is going to actually want to stick with the higher solute potential, so it's not that. The water potential over here is actually a little bit further away from being in term. Well, actually, hang on here. This is that? Hang on. No, 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 that's negative water potential. Okay, so I thought that was a positive there for a second. I was like, oh my God. No, so in other words, the water would not be induced, the water potential itself would not be inducing it to flow over because it needs to be closer to the solute over here. It's not going to want to go over here. So we are actually going to be looking at the pressure potential. And if you notice here, the pressure potential that is here is actually a positive potential, whereas here it is a negative potential. And I want you to remember back to our last lesson where we talked about the fact that there is a negative potential or negative tension happening within xylem. That's why xylem has to have those rigid lignin walls, right? It has to be able to withstand incredible tension force, which that's basically what the guy in the video was saying. It's almost like tension in a spring, right? So we're going to have an actual a pressure differential here that is going to cause the water to flow back here into the xylem. So if we're looking at the forces, again, here we're going to have a solute potential and a water potential that's going to be sending the water from here to here. Higher solute and lower water potential. Higher water potential over here flows from high to low. Over here we're going to have a solute and pressure potential, so higher solute and higher pressure to lower solute and lower pressure, and then we're going to go over here from being a higher pressure over here to a lower pressure over here. Okay, so these are going to be the ways in which we're going to see this water flow and help out the potential of the sucrose as it moves through these lines. Okay, so I hope that's helped you guys to understand how water is going to be working together with the sugar in order to move it through the phloem. Go ahead now and see if you can tackle the rest of the database questions concerning that. The last thing I have to talk about is how we could measure translocation rate. So how could you actually calculate translocation rates? So translocation rates are measured actually using aphids. So I think it's kind of funny that we use aphids for these studies, especially since IB is like, we shouldn't be picking on bugs or other living things, and yet here we are picking on bugs. So aphids are insects that feed on sap through these little teeny tube-like mouthpieces, and they're going to, of course, be sucking up the sap at various different parts along the stem. And so how we use them is we inject radioactive carbon into the plant, which again, carbon is going to be integrated into the sugars. So if it's radioactive carbon, it's carbon in the sugars that we can actually detect and trace. And that carbon is going to be traveling, of course, from the photosynthetic source down to the sinks, and we place a bunch of aphids along the stem, and then we collect the aphids to see what their distance is from the plants and how much time it took from the injection of the carbon into the leaves for it to actually get down to the aphids. And using this, we can actually look at the rates of translocation flow through plants, which is going to be one of the things you're going to be looking at in your database questions. All right, guys, that's all that I have for you for topic 9.2. I hope you guys enjoyed this video. I hope it's helpful, and please email me if you have any questions or concerns. I hope this has been illuminating. Take care.